Welcome to the Generation of Lymphocyte Antigen Receptors module. In this talk, we're going to learn about the diversity observed in the B-cell receptor. We will first learn about two theories, which are the germline theory and the somatic diversification theory. We will then learn about the different gene segments that are present in the DNA that codes for the B-cell receptor, followed by how these gene segments are rearranged to form the variable domain of the heavy or light chain of the B-cell receptor. It is estimated that the human genome codes for about 30,000 genes. However, when we look at the different types of proteins that are present in cells, we have more than hundreds of thousands of proteins that are expressed. This diversity in proteins can be attributed to alternative splicing. When we look at the B-cell receptor and the T-cell receptor proteins, there are different types of B-cell receptor and T-cell receptors that have unique antigen specificities. When we try to quantify how many B-cell receptors and T-cell receptors are there that are able to bind to unique antigens, we have more than 10 to the 11 BCRs and TCRs. That is a huge amount of diversity. And in order to explain how this diversity has come about in the case of immunoglobulins, two theories have been put forth. The germline theory states that each immunoglobulin is encoded by a separate gene that is inherited. Now, this was proposed many years ago, and it is a little difficult to envision that we will have one immunoglobulin that codes for one gene, as we now know that we have a limited number of genes in our genome. Another theory that was put forward years ago is the somatic diversification theory. And this theory states that the variety or diversity of immunoglobulins that has been observed is generated from a limited number of sequences in the V region that undergo changes. As more and more studies were performed, it was found that the DNA sequence that codes for the variable domain of the immunoglobulin is actually made up of relatively small groups of inherited gene segments. So if we look at the light chain of the immunoglobulin, we have two domains, the variable domain and the constant domain. If we concentrate on the variable domain and look at the gene sequence that codes for this variable domain, what we can observe is that that region is made up of two types of gene segments. So here we can see the light chain locus for the lambda light chain. And what we can observe is that there are these gene segments that are called as the variable gene segments. And there are around 30 variable gene segments that are present in that lambda light chain locus. But we also have another gene segment which is called as the J or the joining gene segment. And thus, when we look at the light chain locus of the lambda light chain, we're able to see the presence of two gene segments, the variable and the J or joining gene segment. The multiple V and J gene segments that are found in the light chain gene locus of the immunoglobulins are inherited. Thus, in the lambda light chain locus, the 30 V gene segments that are observed and the 4 J gene segments that are observed are inherited. Similarly, the kappa light chain locus also has V and J gene segments. It has about 38 V gene segments and approximately 5 J gene segments. Now, these numbers can vary from person to person. The heavy chain also shows the presence of gene segments. However, it shows the presence of 3 types of gene segments instead of 2. We are able to observe the V gene segments, and there are approximately 40 V gene segments. But we also have the D gene segments, which stand for the diversity gene segments. The diversity gene segments are not present in the light chain locus. There can be anywhere from 1 to 23 D gene segments, and this depends from person to person. In the heavy chain locus, the J gene segments are also present, and there can be anywhere from 1 to 6 J gene segments. The gene segments that are present in the light chain have to be rearranged in such a way that one V gene segment is joined to one J gene segment. This process is accomplished through DNA recombination, and since this happens in somatic cells, it's called as somatic recombination. The specific name for this type of recombination is called as VJ recombination. So when we look at the germline DNA and look at the light chain locus, we know that there are different V gene segments that are present, and there are different J gene segments that are present. Now, during the recombination process, one V gene segment, so any one of the 30 V gene segments, will be chosen and joined to any one of the 4 J gene segments that are present in that light chain locus. By this joining, we're able to see the DNA sequence of the V gene segment being joined to the J gene segment, and that is what will give rise to the V domain of the light chain. Once the VJ region has been joined through recombination in the light chain, we can see that following that region is the C exon. The C exon is the part of the light chain locus that will give rise to the constant domain of the light chain. It should be remembered that the C region does not undergo DNA recombination. It is only the VJ gene segments that undergo somatic recombination. Once the recombination has occurred in the DNA will be transcribed to form the primary transcript RNA. The primary transcript RNA will then undergo splicing so that the VJ region will then be joined to the C exon. Thus, we see the VJ exon being joined to the C exon in the messenger RNA, and that transcript is the one that will get translated to form the polypeptide chain, which will form the light chain of the immunoglobulin. The heavy chain locus that gives rise to the heavy chain of the immunoglobulin also has gene segments similar to the light chain. However, it has 3 gene segments instead of 2 gene segments that is observed in the light chain. In the heavy chain locus, we have the V gene segments, the D gene segments, and the J gene segments. The D gene segments are the extra set of gene segments that are present in the heavy chain locus of an immunoglobulin, but not in the light chain locus. The heavy chain locus will also undergo DNA recombination the way the light chain locus undergoes DNA recombination. The first step in this recombination event is the joining of one D gene segment with a J gene segment, and this is the DJ recombination event. Once the DJ recombination event is completed, one V gene segment will be joined to this DJ segment, and thus we see the VDJ recombination event. Followed by the VDJ region, we are able to observe the C region, which has the exons that will code for the constant domains of the heavy chain. These constant domain exons will not undergo DNA recombination, and thus the DNA recombination is limited to the VDJ gene segments that will ultimately give rise to the variable domain of that heavy chain. Once VDJ recombination has occurred, the DNA will be transcribed to form the primary transcript RNA, where we can see the VDJ exon followed by the C exons. The primary transcript RNA will then undergo processing, where the exons will be joined and splicing will occur, and then this messenger RNA will be translated to form the polypeptide chain that will correspond to the heavy chain of the immunoglobulin. DNA recombination occurs in the heavy chain and light chain locus of the B-cell receptor in a region that corresponds to the variable domain of the heavy and light chains. We also know that in the variable domain of heavy and light chains, we have the complementarity determining regions. Now, these complementarity determining regions can be mapped to the genes of the heavy and the light chains. And when we look at where they are present in the genes, we're able to see that 2 of the CDR loops can be found in the V gene segment. The third CDR loop, on the other hand, can be found at the junction of the V and J gene segment in the light chain, and in the case of the heavy chain, it can be found in the junction of the V, D, and J gene segments when they are joined together in the case of the heavy chain. When we look at how many gene segments are present in the light chain and heavy chain locus of the immunoglobulin genes, we observe that these numbers differ from person to person. So different people can have different variable or V gene segments, different diversity or D gene segments, and different joining or J gene segments. It should also be noted that not all gene segments are going to be functional, because we do also have pseudogenes, which are genes or gene segments in this case that are not going to be functional due to the presence of mutations. With this, we come to the end of our talk, where we learned about the germline theory and somatic diversification theory that try to explain the diversity that was observed in the case of immunoglobulins. We also learned about the different gene segments that are present in the DNA loci for the BCR, which includes the heavy chain locus and the light chain locus. We also looked at rearrangement of these gene segments to form the variable domain of both the light and heavy chains of the B-cell receptor.